Chapter 11, the lungs, Part 1. The lungs have the central and the critical function of oxygen intake and carbon dioxide removal that sustains life. The lungs diseases that are common are asthma, which affects 8.2% of the adult population. The greatest contributors to lung disease burden are chronic obstructive pulmonary disease and asthma. Chronic lung disease can impair the quality of life and reduce the individual's functional status. These are some concepts for the mechanical and gas exchange function of the lungs. To maintain homeostasis and accomplish movement, as well as cellular work, requires energy. The energy is in the form of adenosine triphosphate, or ATP, which is derived from daily nutrients like sugar and fats. The energy is released in the processes of both anaerobic and aerobic metabolism. The metabolic processes generate carbon dioxide, which is a gas that can cause acidosis, and it must be continuously eliminated by the lungs. At rest, an adult may consume 3.5 milliliters of oxygen per kilogram per minute, or roughly 250 milliliters per minute for a 70 kilogram person. Typically, carbon dioxide production is about 80% of oxygen consumption. During exercise, the oxygen consumption will increase to roughly 10 to 15 times the resting level. Additional lung functions include acid-base balance, immune surveillance, and protection from inhaled pathogens, metabolism of endogenous and exogenous molecules, and contribute to communication in the form of spoken language. The average respiratory rate is 10 to 16 breaths per minute for an adult, with an average tidal volume of 500 milliliters at rest. Air movement into the lungs and out of the lungs is generated by a sequence of contraction and relaxation of the major muscles of inspiration, creating movements of the chest wall and diaphragm surrounding the lungs due to the negative intrapleural pressure. These are various lung disorders that occur across the lifespan and will be discussed in length. The air enters and exits via the nose and the mouth. On inspiration, air is drawn in primarily via the nose, moving successfully through the upper respiratory tract, which includes the pharynx and the larynx, followed by the progressive branching structure of the lower respiratory tract, which is the trachea, bronchi, bronchioles, alveolar ducts, and alveoli. Each level of branching or generation is smaller in diameter than the previous level, but the total cross-sectional area increases dramatically. Beginning with generation 17, as seen in the image, alveoli are present, budding off from the walls of the respiratory bronchioles and permitting the gas exchange. Each alveolus is surrounded by a capillary network which allows rapid exchange of oxygen and carbon dioxide. Each lung has 200 to 300 million alveoli and receives about half of the cardiac output. which is the output of the right ventricle via the right and left pulmonary arteries. The major muscles of inspiration are the diaphragm and the external intercostal muscles. Contraction of the diaphragm requires activation of the phrenic motor neurons that are found at the cervical spinal levels of C3 to C5. The axons of the neurons travel in the right and left phrenic nerves to innervate the diaphragm. The external intercostal muscles, which are innervated by the motor neurons in the thoracic spinal cord, will contract. This will cause the ribs to elevate and move outward, increasing the circumference of the chest. The increase in the thoracic volume pulls the lungs to a more open position and drop the pressure in the airways and the alveoli. When strenuous exertion increases body oxygen consumption and carbon dioxide production, the respiratory rate and the depth increase. During inspiration, the contraction of the inspiratory muscles stores potential energy in the elastic tissues of the lung. The lung elastic recoil converts the potential energy to kinetic energy and contributes to this positive pressure that drives the expiration of the tidal volume. The internal intercostal muscles pull the ribs down and narrow the rib cage and the thoracic cavity. This compresses the lungs from the circumference. The combined movements create positive intrapleural pressure that translates to positive pressure inside the airways, increasing the expiratory airflow. The neural control of respiration is initiated within the brainstem, primarily from a pattern generator that influences clusters of medullary neurons controlling the inspiratory muscles. Inspiration ends when the medullary inspiratory neurons cease firing. This decreases the drive to the motor neurons and allows the inspiratory muscles to relax, with expiration occurring passively. The respiratory control system is pathologically disrupted by neurological conditions like stroke, brainstem or high spinal cord injury, lower motor neuron lesions of the phrenic nerve due to Guillain-Barré syndrome, polio, or prescribed or even illicit street drugs that suppress the respiratory centers like opioids, alcohol, benzodiazepines, and barbiturates. As the airways repeatedly branch from the trachea to the alveoli, the smaller segments begin to have alveoli capable of gas exchange. The transition from conducting airways to respiratory airways begins at about generation 16 to 17. The upper airways have mucus-producing goblet cells and submucosal glands. The lining epithelial cells are ciliated. The protection from inhaled particulate matter is achieved by mucociliary clearance. The smooth muscle cells are responsive to autonomic nervous influences, producing dilation under sympathetic control or constriction under parasympathetic control. The elastic fibers contribute to the elastic recoil that drives passive expiration. Air is inhaled first into the upper respiratory structures, beginning with the nose. The inspired air is warmed and humidified. The mucus lining of the nasal passages begins to trap particle matter before it can enter the lungs, and the cilia move the mucus toward the oropharynx. The pharyngeal structures include the tonsils and the adenoids, which are lymphoid tissues that are able to conduct immune surveillance of the air that is entering the airways. Beginning with the trachea, the airways are generally divided between the conducting zone that begins with the strong cartilage reinforced trachea and progressively divides into smaller bronchi and bronchioles, and the respiratory zone, which consists of small bronchioles with alveoli budding off and ending in alveolar sacs appearing like clusters of grapes. In the conducting zone, the walls have abundant goblet cells that produce and secrete mucus. The ciliated epithelial cells have numerous cilia that beat in one direction toward the mouth, continually moving the mucus layer to the mouth, from which it then can be swallowed or expectorated. As the airways get smaller, they have less or no supporting cartilage, with two significant consequences. First, the airways are prone to collapse if the pressure surrounding the airway is greater than the pressure inside the airway. Second, the bronchial smooth muscle contraction is able to narrow the collapsible airway. Mucosal dendritic cells phagocytose and present antigen to T lymphocytes, activating the adaptive immune system to produce secretory immunoglobulin A. IgA provides protection against specific invaders by interrupting incoming pathogens and marking them for phagocytosis and destruction. Alveolar macrophages secrete cytokines that recruit additional monocytes, neutrophils, and lymphocytes to assist in host defense. Natural killer cells are present in the alveoli and can contribute to a rapid response to invading pathogens. Cystic fibrosis is a common autosomal recessive genetic disorder among certain populations. There are close to 2,000 mutations in the gene coding for cystic fibrosis transmembrane conductance regulator, or CFTR channel, a large transmembrane protein found in cell membranes that permits the movement of chloride and bicarbonate across the epithelial membrane. This F508del mutation results from a deletion of 3 nucleotides that code for a phenylalanine at amino acid production 508 within this very large and complex protein. The result is a misfolded protein that accumulates in the endoplasmic reticulum and is targeted for degradation without ever reaching the plasma membrane of the cell. The pathophysiological consequences of CFTR mutations are manifested in tissues that depend on a lining layer of mucus that can support movement of secretions along tubular structures. These mutations are associated with chronic lung disease, diminished pancreatic enzyme secretion leading to poor digestion and nutrition, abnormalities of bile secretion, and infertility. Without normal CFTR function, the mucociliary clearance mechanism is severely impaired. A lack of chloride transport across the epithelial cell membranes reduces the associated water movement, so the mucus layer is abnormally thin and extremely viscous. The loss of bicarbonate secretion makes the mucus acidic, reducing the effectiveness of white blood cells in attacking pathogens. The function of the associated sodium ion transporter is increased, promoting intracellular sodium accumulation and water retention, which contributes to thick mucus.